Experiment 26 in Chem 1212 is titled Redox Titration of a Metal Oxalate Compound with Permanganate. And in this video, I want to do two things. Firstly, I want to revisit the concept of titration and really get to the heart of what it means to do a titration and what the essential elements of a titration are. It's not as simple as acids and bases, and this is a good thing. Titration is a very versatile technique. The second thing I want to do is revisit redox reactions and talk about the specific redox reaction that we'll rely on in this experiment. We're going to perform both a standardization and a titration with the ultimate goal of figuring out the number of oxalate molecules per molecule of metal oxalate that was synthesized in experiment 25. So let's begin by revisiting the concept of titration. Titration does not depend on acid and base reactions specifically. To emphasize this, I want to work with a general reaction A moles of A plus B moles of B goes to C moles of C and D moles of D. Now the typical situation in a titration is we have some sample that has an unknown amount, an unknown number of moles of one of the reactants A in it. And because we're measuring it, we know the amount, the number of moles of another reactant. Because of the stoichiometry of the reaction, and typically a visual or quantitative indicator of some kind, we also know the amount of at least one of the products that's present as we add that reactant B to the reaction mixture. The unknown species that's part of the sample we call the analyte, and the known reactant that we drip into the analyte we call the titrant. And we know its amount because we know its concentration, which is moles per volume, and we're measuring it dropwise, so we're measuring the volume as we add it. Now the concentration of one of the products, let's call it C here, is also known typically through visual means, so we can at least know when we've reached a special point where all the reactants have been consumed through visual means or through the use of something like a pH probe. And from the stoichiometry of the reaction, we can figure out how much product is present, although that isn't really directly related to calculating the amount of A present. This is the typical setup of known and unknown variables for every titration. And the important thing I want you to take away is that any species could be involved in this reaction. But what are the conditions on a reaction that's used for titration? Well, there are a couple of important ones. First of all, the reaction must be complete. This means that thermodynamically it heavily favors the products. If the reaction favored the reactants, then we couldn't be certain that adding B was going to induce the reaction to occur. That's knowledge that we've got to have if we want to use a reaction for titration. The reaction has to heavily favor the products. And it's also beneficial if it's rapid. Now that's not a strict requirement, but the more rapid the reaction is, the less time we have to spend on the titration itself. Any species A can be measured through titration. We're limited only by our ingenuity and creativity in finding or coming up with complete reactions of the species A with some other reagent B. And briefly, it's worth mentioning that the titrant is usually something that is very reactive. You'll notice that if you look back through titrations we've done already. You titrate typically with a strong acid or a strong base. The reagent is strong, which means that we can be assured that reaction to the right is going to occur to a very large degree. For the specific system we'll be investigating in experiment 26, we'll be using potassium permanganate, KMnO4, as the oxidant. And it's a strong oxidant. It's a very reactive species. So KMnO4 will be our titrant, and unsurprisingly, it's something that very completely oxidizes a whole lot of substrates, including oxalate. One of the Lewis structures for the permanganate anion is shown for you here, and if you examine the manganese atom and calculate its oxidation number, you'll find that remarkably this is manganese in the +7 oxidation state, which is huge. This manganese is dying to oxidize something so that it can lower its oxidation number. Under acidic conditions, it reacts with numbers of protons and electrons that I'll leave up to you to figure out, to form manganese 2+ and a certain number of water molecules. So if you haven't done this already, I strongly encourage you to balance this reaction on your own. And notice that the manganese atom is manganese 2 in the products. Let's talk a little bit about the oxalate dianion, which turns out to be a mild reductant. The Lewis structure of oxalate is shown for you here, and oxalate can form 2 molecules of carbon dioxide, which are gaseous and can float away, and it can release a certain number of electrons in the process. Again, I'll let you fill in the exact number of electrons here. This corresponds to an oxidation of the oxalate and a reduction of whatever receives those electrons, which in our case will be permanganate. In the standardization phase, we're going to calculate exactly, with a high degree of precision, the concentration of permanganate anion in the KMnO4 solution that we'll use as the titrant. The basic idea is similar to standardizations that we've done already. We're going to start with a burette of KMnO4 and an aqueous solution of sodium oxalate as the so-called analyte. And we're going to drip KMnO4 into that analyte solution until the pink color persists, and we'll talk about why we do that in a second. So the sodium oxalate is what's called a primary standard. It's something that we know the composition of, we know the mass of because we've measured it, and because we know the molar mass, we also know N, the number of moles of sodium oxalate present in that solution. The titrant is a solution with unknown concentration of KMnO4, but we can calculate the number of moles that we've added once that pink color persists from the stoichiometry of the reaction and the fact that at the instant we see color hanging around in the analyte solution, we know that we've used up all of the oxalate. All of it has reacted. The volume of titrant added to this point is measured, and so we can calculate the precise concentration of permanganate by dividing that number of moles that we got from the reaction stoichiometry and knowledge of the endpoint by the volume added, which is measured. You'll see that in the titration phase, we need this precise concentration to be able to figure out the number of moles of oxalate anion present in our unknown analyte. So it's critical here that we know everything about the analyte in the standardization phase, and what we don't really know is the volume of titrant necessary to react with, we would say in an acid-base titration, neutralize all of the sodium oxalate. Now let's discuss the titration phase of experiment 26. Although we will need some information from the standardization phase to do calculations related to this stage, in the laboratory you can carry out the standardization and the titration simultaneously. And in fact, the setup is identical to the setup for standardization. We have a burette of KMnO4 solution, and now, at least theoretically, because we've done the standardization phase, we know the exact concentration of permanganate anion. And we can calculate the number of moles of permanganate that we've added via the volume, which we'll measure, using the fact that the number of moles is linearly proportional to the volume, and the factor of proportionality is the molarity of permanganate. The analyte is now different. The analyte now is our unknown oxalate, which contains some number of oxalate molecules X per molecule of the entire complex. And at the top here, you can see I've abbreviated this as MC2O4X ... to indicate that there could be other things present. What we're really interested in is the number of molecules of C2O4 per molecule of the entire complex. The value X is unknown, but we do know the mass of complex that we added to the water to prepare the analyte solution in the first place. And that's going to become useful when we end up trying to find X down the line. So just like we did for the standardization phase, we're going to drip the KMnO4 solution into the analyte until we observe a pink color persisting. That means we've consumed all of the oxalate present in that complex. Notice that this assumes a couple of things. It assumes that the oxalate dissociates completely from the complex and that reaction with permanganate is complete. You won't have to really worry about these assumptions, but it's worth pointing them out. We're assuming that all of the oxalate gets consumed as we add permanganate, right up to the point when that pink color persists. That means we've got an ever so tiny excess of permanganate ion present. Because we know the concentration, we know the amount in moles of permanganate added, and we can use the stoichiometry of the reaction to determine the amount of oxalate that was present in the analyte solution. So we're getting closer to what we want. We know at least the number of moles of oxalate that were present in the analyte solution. But we don't know the number of moles of the complex that we started with, right? Because the molar mass of the entire complex depends on X. So we can calculate the mass, at least, of oxalate anions that were added when we added the entire complex into the water to create the analyte solution. And from that, we can calculate a mass ratio or a mass percent of oxalate in the complex. And this is the mass of oxalate, which we calculated from the amount of oxalate, which was in turn calculated from the amount of permanganate, divided by the total mass of the complex, which depends on x, but we can at least plug in hypothetical values of x, 1, 2, 3, etc., and generate theoretical mass percentages that oxalate should have if x is 1, 2, 3, etc. And you'll know this if you look back at experiment 25. The molecular formulas of the products are given there, and so you can generate a theoretical estimate for the weight percent. Compare the results of your weight percent to the theoretical given in the lab manual, and if it's close, you'll know that you've done the titration correctly.